Hello and welcome to our presentation on engineering functional and histological regeneration of vascularized skeletal muscle. Next slide, please. So what's the issue? Volumetric muscle loss, also known as VML, is a defect that impacts greater than 20% of muscle mass, and it can't be repaired by the body's repair systems. So it leads to functional deficits because the muscle can't perform as well as it would if all the muscle was present. Current treatments for VML are really limited, so things such as autotransplantation are really limited by the availability of donor tissue and something called donor site morbidity, which is complications at the site where the muscle was taken from. Tissue-engineered grafts have emerged as sort of a solution to this issue, so they have the possibility of creating greater clinical options for treating VML because you don't actually need a donor, you can just grow the tissue independently. Next slide, please. Decellularized extracellular matrix, also known as DECM, both acellular and with cells, have been explored as a possible scaffold for VML treatment, but this model as well has had many issues. So acellular DECM can't spontaneously generate myofibers or muscle fibers, and then the cell-seeded DECM is prone to batch-to-batch variability, and it's also prone to provoke an immune response from the body. This experiment specifically used electrospun fibrin as an alternative to the DECM, and because fibrin has so many properties, such as being biodegradable and FDA approved, that makes it an ideal candidate for skeletal tissue engineering. Next slide, please. So this experiment specifically tested axially aligned fibrin hydrogel microfiber bundles and their ability to support the development of skeletal muscle constructs using C2-C12 myoblasts. So what that means is basically they were testing the ability of the scaffold to promote skeletal muscle growth from C2-C12 myoblasts. Myoblasts are a muscle fiber or a muscle cell that has the potential to grow into a muscle fiber, and C2-C12 is just the mouse strain that it was taken from. The reason we chose, the experiment chose fibrin hydrogel is that the elastic modulus is very similar to that of skeletal muscle. They also tested the ability of the hydrogel with and without C2-C12 myoblasts to treat VML, specifically in cases where 30 to 50% of the muscle fiber was removed. Next slide, please. So this experiment hypothesized that the combination of the C2-C12s on the electrospun hydrogel scaffold would promote muscle regeneration and functional recovery greater compared to the scaffold alone or no repair.
Thank you, Alex. Hey guys, I'm Joe, and we're going to be presenting the methods and techniques for this experiment, starting with the scaffold itself and the myoblast integration. So looking at the electrospun hydrogel with the fibers as the scaffold, as mentioned earlier, the electrospinning fibers adds the structural element that we're looking for the scaffold, and the hydrogel allows the cells to proliferate and expand throughout the region as necessary. And these properties are tunable both chemically and mechanically, and we perform tests in the experiment to figure all these out. One example of this is testing elastic modulus using a bioreactor. And then another method was the cell culture and seeding of the actual scaffold. We used C2-C12 myoblasts, which were pipetted into the scaffold, and these were incubated and then used to help in the muscle regeneration. Moving on to the next slide, we'll see an image that depicts how the electrospinning actually creates bundles of fibers which are advantageous to the structure of the suture and the scaffold. And then we look at the image below where you can see the cells are seated within the electrospun fibers. Moving on to the next slide now, we're going to look at some other techniques. This is the immunostating of the scaffold, where you can track the movement of the myocells throughout the fibers. As you can see here, primary antibodies bind to either the fiber or the myocells, followed by the secondary, and finally the color. And we use DAPI to visualize the myocells, which are shown in blue throughout the fibers there. And we use MHC red to stain the actual fibers themselves, which can be visualized with the microscope. Moving on to the next slide, we look at assessment of the scaffolds themselves and testing the properties, testing how they perform. We look at in vitro measurement, which is outside the mouse, of course, shown in the bottom left. We allow the scaffold and the seeded scaffold to grow in a plate, and then we measure the force, the contractility force, after electrical stimuli have been applied. And this can be shown here. We attach the electrodes, apply electrical stimuli, and measure how much it can contract. Next, we look at the VML defect model shown in the bottom right, where we remove muscle from the actual leg of a mouse, and then implement our solution, which would be the scaffold, allow it to grow, and then assess it after 2 and 4 weeks. Moving on to the next slide, we look at the histology of the actual muscle removed after this 2 to 4 week period, and we check to see how the myocells have spread, how the fibers have spread, using the method mentioned earlier, which is primary, secondary, and finally the color itself. There are some other alternative methods mentioned there, including Mason's trichomy and H&E staining as well. Now we move to the final slide for methods looking at the assessment of scaffolds in vivo using functional testing, which would be attaching the mouse to an actual device to measure the force of contraction of the muscle. And we, of course, apply electrical stimuli to stimulate that force. And we look at statistical analysis to evaluate the difference of groups in these different tests and to see how significant they are. Now we look at the results.
Thanks, Joe. Okay, so in terms of the scaffold alone, we found that the C2C12, which are the muscle cells seated in the scaffold, looked the most like mature muscle, and this is indicated by the multinucleation, which is cells with multiple nuclei, and the sarcomeric striation, which are aligned muscle fibers, and these are two key elements of natural muscle cells. The C2C12 is cultured just on a glass plate without the scaffold, lacked an alignment, they were randomly assorted, and they did not exhibit the multinucleation or sarcomeric striations. Both the bulk and spread scaffolds, which we'll see on the next slide, which are different configurations of scaffolds, resulted in significant muscle regeneration. And due to this similar performance, bulk scaffolds, which are easier to produce, are used in the future experiments. So moving on to the next slide, you can see the bulk versus spread scaffolds. The main difference is just how the scaffolds are spread apart. Moving on, there were three groups compared with regard to the percent of the tibialis anterior muscle regeneration. And this is the untreated, the acellular, and the C2C12, and these were compared to the uninjured values. So the untreated defects continued to lose muscle. The acellular scaffolds lacked the major histocompatibility complex, and that's a cell line which is very important for a successful immune response, and this is so the body doesn't attack the implant. The C2C12 seated scaffolds had significant volume recovery, and they resembled the most native muscle structure. And so you can see in this graph just the different weight of the muscles. So this is a measurement of regeneration, the uninjured, the untreated, acellular, and C2C12, which were significantly different. So this just shows the staining of the various muscles. So the untreated is the smallest, acellular and uninjured are the next, and then the C2C12 had the most growth, and this is stained with the MHC, which shows the immunoline of the cells, and then DAPI, which stains the DNA, so you can see the actual myoblast cells. In terms of mechanical recovery, both the C2C12 and acellular scaffolds had complete functional recovery compared to the uninjured values. There was no significant difference between the seated scaffold and the acellular scaffold in terms of the mechanical response. So the max isometric torque, which is tension only, was measured, and at 2 and 4 weeks, there was full recovery of these values.
All right. So just in summary, this group sought to create a novel muscular and vascular regeneration system based on electrospun fibrin scaffolds and myoblast cell seeding. And in conclusion, the end results showed significant potential for this particular scaffold design, one which the team believes can be used to fully recover muscular structure and function lost in VMLs. This potential treatment option is particularly exciting and promising due to the past limitations that it had overcome, including but not limited to low myofiber and vascular regeneration upon integration of the scaffolds, something that was extremely problematic for acellular scaffolds, which were able to regenerate, as Seth said, functional contractility, but not the actual myoblast synthesis. Other limitations include high fibrosis or scarring at the graft-host interface and poor cell viability, which both hindered myoblast integration and growth with the native skeletal muscle. Another huge limitation was poor biocompatibility, excessive immune response, which hindered further growth of the new muscle mass. So this system was able to overcome most of these limitations by mimicking skeletal muscle mechanics, stiffness, robustness, density, and contractile function, and alignment cues, with little to no fibrosis. That was the big thing. It also improved vascular filtration, biodegradability, biocompatibility, and suturability. Next slide, please. In terms of next steps, yes, this is certainly a potential treatment for VML. There's also opportunities for scale-up testing using larger animal models so that it can hopefully be put on a track to be used for human VML patients. Future studies might also be influenced based on this seated scaffolding model in implantable vasculature, and further studies might be done on the contribution of ASCs to collagen deposition in muscle defects. Next slide. In terms of overall thoughts, acellular biological scaffolds, extracellular matrices have long been thought of for potential treatment options for VML, but have not been successful, mainly due to two issues. One, biocompatibility, and two, integration. This system stood apart from the rest because they actually focused on those two big factors by using their cell-seeded model. This study showed our group that without a biocompatible material and a structurally sound mechanical design, one cannot actually create a successful implantable scaffold. So thank you very much.